All right, so welcome back to the NIH Library's Bibliometric Training Series. My name is Chris Belter, and I will be your instructor for this course. So with this course and with the next couple of courses in this series, we're going to be making sort of a cognitive shift towards network science and looking at the applications of network science in bibliometrics. So this first course will be talking about collaboration networks, specifically co-authorship networks at individual levels and also organizational levels. The next course is going to be all about visualizing networks using an open source tool called Gephi. The next course after that is going to be all about cleaning those visualizations up, working with them in a vector editing software tool, which I'll explain what that is when we get there. And finally, the last one in the series is going to be about topic analysis using networks in various ways to get at what topics are all about. So again, all of these are going to be related to networks in some way. They're also going to be picking up where the previous one left off. So with this first one, I'll sort of go over how to actually create networks in the Science of Science tool. The next one is going to pick those networks up and actually go into how to actually visualize that network. Then the third one is going to be about taking the network that you created in Gephi and then working with it, cleaning it up, working with it in Inkscape. And then the final one is going to be kind of a refresher around some different applications of these things, and then sort of looking at combining these things to do topic analysis. So again, each one of them is going to build on the previous week's lesson. So it's really important to finish each one before moving on to the next one, because they're intended to be viewed as a series. At least the first three of them are, because you'll need to know what we covered in the previous session in order to be able to start with the next session. So that's kind of what we're going to do. So again, this one is all about actually creating networks, and specifically collaboration networks in the Science of Science tool. So what we're going to do is we're going to start off by giving you sort of a crash course about network science and about networks in general. What we're talking about, a brief introduction to some of the lingo of network science, and then also go over a little bit about some properties of networks that are really important in doing bibliometrics. Again, this is not going to be comprehensive by any means, but it's at least an introduction so you know what's going on. So we're then going to talk a little bit very briefly about the Science of Science tool itself, which is the tool that we're going to be using for creating these networks. And then I'll jump into a live demo in creating both individual level co-authorship networks and then organization level co-authorship networks, both using the Science of Science tool. So let's start out by talking a little bit about networks and network science. So network science is a very big, very rapidly moving field. It's also a very interdisciplinary field. A lot of it comes from physics, some of it comes from computer science. It comes from a lot of different places. And this is a very big, active field of research, so there's no way that I can cover everything that you need to know about networks here. This is really just a primer to sort of get you thinking about it, get you understanding what it is that we're actually dealing with here, and again, introduce some of the terminology that is going to be used in network science. The reason that I'm doing this here is because a lot of the tools, like the Science of Science tool and like Gephi, assume that you know the lingo. They assume that you know what a node is. They assume you know what betweenness centrality is. They just assume that you know what these things are, why you would use them, those kinds of things. So I wanted to give you a quick introduction before we actually get into any of the actual using things, so that you've got some of the basic understanding of what that language and terminology is. So it's at its most basic level, a network is just a set of relationships between things. So with network science, we're not as interested in the individual things as we are in the relationships between those things. So we're interested in how things connect to one another, or how things move from one thing to a different thing, or in some way how things relate to one another. So we're interested in that both at the sort of the individual element level, but also at the macro level. So we're looking at the entire structure of these set of relationships and what it can tell us about the actual network itself. So we're as interested in the sets of relationships as we are about the individual things that come out, because there's a lot of things related to the structure of these networks that become really important for understanding the things that we're actually looking at. Within a network is really composed of two very basic things. The first of those things are called nodes. Sometimes in computer science, they're also or in physics, sometimes they're also referred to as vertices. Sometimes they're also referred to by other names, but in bibliometrics, we tend to talk about them as nodes, so that's how I'll refer to them throughout these series. So the nodes are the actual things in the network. So these are the actual elements that we're looking at relationships among. So these nodes can have a number of attributes associated with them. So they can belong to certain categories, or they can have certain numeric attributes associated with them. They can actually have a whole range of these attributes depending on what kind of network we're looking at. But again, these nodes are really just the things that we're looking at. They're the individual components of the actual network. The second part of the network are called edges. I have no idea why they're called edges. They just are, so just roll with it. These edges are essentially the connections between nodes. So if there's an edge between two elements within the network, that's called an edge. And so that edge indicates some form of relationship between the two things that are connected. So these connections are, again, sometimes they're called connections, sometimes they're called edges, sometimes they're called links. They're called a lot of different things. But again, within the context of this course, I'll be referring to them as edges. So like nodes, they can have a number of different attributes, but they can actually be much more complicated than nodes are, because there are certain kinds of attributes that are referred to in different ways. So edges can have a direction associated with them. They can either be directed or undirected. Essentially what this means is that in the network that we're looking at, something moves from one node to a different node over that particular edge. So in this case, think of the electrical power grid as a network. So the individual nodes would then be power stations, transformers, and then individual houses. And then the edges would be wires that would connect the power station to the substation. Those would be directed because there's a direction in which the electricity flows from the power station to the substation to the individual person's house. So that would be an example of a directed network where something moves from one node to another node in a particular direction, but not necessarily in the opposite direction. So when you see networks referred to as being a directed or undirected network, that's what they're talking about. Is if it's a directed network, there is some kind of an arrow pointing from one node to another node. If it's an undirected network, there's just some sort of relationship. There's no actual direction associated with the edges. The other types of attributes that can be associated with edges are, edges can be called weighted or they can be unweighted. And this just basically means that there's some kind of a numeric attribute associated with the edge. So again, think back to that electrical power grid for a minute. We can actually measure the amount of electricity that goes from a power station to a substation. And so that would be, there's an amount of electricity that flows in a particular direction. So that would be a weighted network because there's an amount of electricity that goes from one node to another node. In other cases, there may just not be any quantitative value associated with the edge, in which case they would be referred to as an unweighted network. So we can actually combine these, and we can talk about a network as being a weighted directed network, which means basically that there's a direction associated with the edges, and there's a quantitative attribute also associated with those edges. So we can get a lot of information about a network by sort of some combination of these things. So we can look at directed unweighted networks or undirected weighted networks, all kinds of different combinations of those things to sort of look at what this network looks like and how it actually works. Edges can also have certain categorical attributes associated with them. We don't really see that very much in bibliometrics, but we do see that a lot in other kinds of networks, particularly in bioinformatics networks. There can be particular kinds of edges associated with particular kinds of networks. That's getting a little bit more complicated, but I just again wanted to point out that that is a possibility if you're interested in moving in network science in that direction. But for the most part, when we're looking at bibliometric networks, we're looking more at just sort of edges. Sometimes they're directed, sometimes they're undirected. Most of the time they are weighted, but sometimes they can also be unweighted. So it just depends on the kind of network that we're actually working with. So when we're actually going to look at a network, this is what a network might look like in terms of what a visualization of a network might look like. So nodes, or again, are these elements of the network. Typically they're represented by circles. Sometimes they could also be squares or other types of things, but typically they're represented as circles. And then the edges, or those relationships between them, are usually represented by lines connecting them. In this case, we're also adding in some additional attributes. So the nodes are actually sized based on some attribute associated with them. So they're larger or smaller based on the quantitative value. The edges are also larger or smaller. They're also darker or lighter, depending on the amount of or the weight of the edge, or the quantitative value associated with that edge. This has all been pretty abstract so far, so let me give you a couple of other examples of very common kinds of networks that we see on a pretty regular basis. One that's probably the most familiar is a social network, where nodes are individual people, and then the edges are whether there are friendships that exist between those people. These can either be in-person friendships, or they could be Facebook friendships, they could be Twitter followers, there could be a number of different things related to this, but essentially we're looking at friendships between people. In this case, sometimes there can be a weight associated with it, and sometimes there may not be. In just a traditional social network, there's usually not a weight associated with a friendship. It just is there or it's not. So in that case, we would call it an unweighted network. It's also usually an undirected network, because presumably if you're friends with somebody else, they are also friends with you. You know, that's typically the way that works, but you know, I don't want to get too far into the weeds there. But typically they are undirected and unweighted networks. In some cases, there may be weights associated with them if you're sort of making this at a higher level of abstraction. One thing that's also pretty familiar to a lot of us is the concept of the 7 degrees of separation, particularly the 7 degrees of Kevin Bacon. So this was all about looking at connections between actors in movies based on whether or not they both appear in the same movie. So one of the things that they found was you can get from Kevin Bacon to just about any other actor with 6 or 7 steps. This is actually by no means unique to Kevin Bacon. This is actually true of just about any actor who's ever appeared in more than one movie. It actually comes out of the actual network structure that emerges in this actor network, and we'll get into why that happens a little bit later on in the talk. Another example of a network would be websites on the World Wide Web. So in this case, the individual nodes would be websites, and then edges would be hyperlinks where web traffic can move from one website to another website. So in this case, this would be a directed network because, again, the direction of the link matters. Because if you link from one site to another site, there's not necessarily a corresponding link back to the original site. So in this case, the directions very much do matter, and this would be a directed network. Most of the time, these are unweighted. So when we're looking at website networks, we're really just looking at whether or not there is an edge or whether or not there is a link from one website to another one. But in some cases, you might be able to actually measure the amount of clicks that click on a particular hyperlink or other things where you could actually build some additional quantitative variables into this kind of a network. Another kind of network that's very, very common in biomedical research are gene regulatory networks. What's interesting here is that the nodes actually represent genes, and then the edges between them represent interactions or regulation relationships between genes. You could also do gene RNA networks, RNA protein networks, protein-protein networks. There are all sorts of networks that come out in biological sciences and particularly in bioinformatics. There are also networks related to neurons in the brain where neurons are connected if there's some kind of electrical signal that goes from one neuron to another one. So there's lots of these kinds of networks in bioinformatics and in biological sciences. Final kind of network that I'll talk about as an example are obviously these co-author networks. And so moving this more into bibliometric realm, this is the primary network that we're going to be working with and looking at today. So in this kind of a network, the individual nodes are authors in a particular publication set, and the edges or the links between them are co-authorships on scientific papers. So with this kind of a network, if two authors write a paper together, that creates a link between those two authors. So this kind of a network would be an undirected network because there's just whether or not there is a co-authored paper or not. There's no direction really associated with that edge. It's just a measure of whether or not it happened. But it can be weighted. So it can be weighted based on the number of papers that two authors write together. So when we zoom out a little bit and sort of start looking at these numbers of co-authorships among people, we can start to get ideas of who tends to work more closely together. We can start to see groups of authors who tend to work together on a frequent basis, those kinds of things. So again, it's just looking at the structure of this overall collaboration network, we can start to get a sense for some of these collaboration patterns. At another level of abstraction, we're also going to be looking at organizational level co-authorship networks where instead of authors, they're actually organizations. So the affiliations of these authors and whether or not people from these organizations actually co-author papers together. In every other way, they're exactly the same as individual level networks, but again, we're looking at, it's a slightly different level of abstraction where the nodes are something slightly different. But again, we're looking at co-authorship as a measure of collaboration in these kinds of networks. One of the things that's been very, very interesting in the development of network science is even though we're looking at these very, very different kinds of networks, there are certain properties that seem to be universal among all of these different kinds of networks. So there's certain properties about networks that are common across all kinds of networks. So these are true of social networks, they're true of paper citation networks, they are true of gene regulation networks, all kinds of other types of networks. As far as we know, these are universal characteristics. So I'll point out a couple of them that are the most important for understanding some of the things that we're going to be doing later on. The first of these properties is this idea that networks are, quote, scale-free networks. This essentially means that some nodes within the network are really, really highly connected. And then most of the nodes in the networks have few connections. So one of the things that we sort of look at is, again, in a co-authorship type of a network, some authors have a very, very high number of co-authors, whereas most authors have a relatively low number. So if you're familiar with mathematics at all, we could say that the number of connections follows an unequal or a skewed distribution, where, again, some of these nodes have a very, very high number of connections, whereas most people in the network have very, very, very few connections. This is also true of the electrical power grid. It's also true of the web. It's also true of gene regulation networks, all kinds of things like that. What's interesting about this is this scale-free property actually arises from another network property called preferential attachment. So what this basically means is that nodes that already have a high number of connections are much more likely to receive additional connections than other nodes in the network. So if there's a node that already has 3 or 4 connections, it is much more likely to get additional connections than it is to any of the ones that was actually originally connected to. So what happens is as networks form, these networks actually build up from a relatively small number of nodes. So when a network actually starts, there's a relatively small number of these nodes, and the nodes are added to the network over time. So as the network grows, there are new nodes added and new connections added to the network. And so the way that these new edges are actually established is through this process of preferential attachment, where essentially the rich get richer. So the more connections you have, the more likely you are to get additional connections. And so this preferential attachment leads to that scale-free property, where it leads to certain authors or certain nodes in the network becoming very, very highly connected and then becoming more highly connected over time. Another result of this sort of network growth property is this idea of the small world network. So this kind of gets back to this 7 degrees of Kevin Bacon idea, where most nodes are very closely connected. So it takes a very small number of steps to get from one node to any other node in the network. So most of the time, with most of these networks, it only takes about 5 or 6 steps to get from one node to another node. Even if you're talking about tens of thousands of nodes in a network, it's still a relatively small number of steps to get from one node to any other node. And this is, again, because of that scale-free property. There are these nodes that are very densely connected and act as sort of hyperconnectors between different parts of the network. So even though 2 authors or 2 actors may be relatively separated over time or space, there are these very dense hubs that are very highly connected that act as connectors to bridge those 2 different sides of the network. The final property that I'll talk about that's important for network science is this idea that networks are modular in structure, or in a sense, networks tend to consist of subnetworks. They tend to consist of a group of subnetworks that are more connected to each other than they are to other nodes in the network. So again, think of the co-authorship idea. There are a number of authors who tend to write papers together, whether they're at their own laboratory or they're just established working relationships between these authors. They tend to stay within those groups and tend not to write papers as frequently with people outside those groups. So within any given network, you've got these very densely connected subnetworks within it. Again, with the internet, we see very densely connected groups of websites that are all around a particular topic. So they all tend to link to each other, but tend not to link to other sites about different topics. This becomes very, very important when we're looking at bibliometric networks, because we're very interested in the ways that people actually interact with each other, the degree to which they actually collaborate with outside of these groups, what these groups are, and so on and so forth. This is also very important when we start getting into some of the topic analysis using network science, because within papers, there are similar kinds of these sub-networks that are more densely connected to each other, either by the words that they use, the references that they cite, or other things that where, again, these papers are more connected to each other than they are to other papers. And so that these groups of papers tend to correspond to research topics or research directions. So many of the topic analyses and analysis methods that I'll be talking about later on really rely on this sort of modular structure. So that's essentially everything that I wanted to cover about network science. Again, there's a ton of additional information about network science that we could go into in much more detail later on. Some of those are in the readings that are associated with this week's course. Other things are available online or in other books, so on and so forth. But for the purposes of what we're doing here and in this series, these are the major ones that I wanted to talk about. So shifting gears now from the network science to actually some of the hands-on portion of this thing, I wanted to bring up a couple of points about the actual tool that we're going to be using in this course for actually creating networks. So the tool that we're going to be using is the Science of Science tool. So this tool is developed by a number of folks at the University of Indiana. It is free, open source, and freely available, so you're welcome to just download it for any purpose and just go ahead and start using it. It has quite a lot of documentation associated with the Science tool. There's an extensive user manual available. Again, all of that is on the links that are available here. Feel free to dig around and have a look. There's quite a lot of information there, so definitely look around. What's interesting about this Sci2 user manual is it somehow manages to be very extensive and somewhat unhelpful at the same time. I don't know quite how they manage it, but that seems to be the case. There's a lot of documentation that is very difficult to understand. It has a lot of network lingo. It has a lot of terminology that can be very difficult for a beginner to understand. So fair warning, it can be somewhat difficult to actually learn the tool from the manual. If you're going to look at this Sci2 manual, I would recommend skipping the vast majority of it and going directly to the workflows. There's a series of workflows for using the tool for creating various kinds of networks. I would recommend going directly to those workflows, try and see if you can work through those workflows and get a sense for how these things work before you start trying to read some of the documentation about the individual algorithms and other things. So go straight to the actual hands-on portions, try and work through those, and then try and start learning about what's actually happening at each step. That would be my recommendation. I also wanted to offer a few quick notes about the installation of Sci2 because there are a number of things that can go wrong in this particular step. So I wanted to sort of give you a couple of sort of lessons learned that we've gotten from working with it, trying to install it on a range of platforms and computers, so forth. The first is the actual version that we're going to be using in this course and that we're going to be using here at NIH. We have found that the most recent version, version 1.3, can be very unstable. In many cases, it just doesn't work on certain platforms. Anecdotally, we've never actually been able to get it to work here at NIH. So in this course and in all of the follow-up courses, we're going to be using version 1.2 beta, which is the older version. It's much more stable. It's, I believe, from 2015. So that's the version that we're going to be using in this course and throughout the courses. You're welcome to try to get version 1.3 to work. If it works for you, great. Feel free to use it. We have never been able to get it to work, so we're using version 1.2 beta. Sci2 is also interesting that it doesn't necessarily actually require an installation for it to work. You essentially can download the zip file, unzip it, and then theoretically you can go ahead and start working with it. However, in practice, we found that it's very picky about the location where it actually is installed. So you essentially need write permissions to the folder where you're actually running Sci2 from. So Sci2, as it works, is actually going to create a log file in the background where it actually keeps track of all of the things that you do in the Science of Science tool in any given session. What that means is it's going to create that file in the same folder where the actual application is installed. So if you can't create or write files in the folder where it's located, Sci2 is not going to work. So for those of us who don't actually have administrator rights to our computer, that means we can't unzip it and install it in our program files folder. That would be the logical place to put an application in the program files folder, but unfortunately, if you don't have the ability to write files to your program files folder, Sci2 is not going to work. So you need to install it someplace else. So essentially someplace where you can write, create, and edit files. In practice, it doesn't really matter where it's actually put as long as you actually have write permissions to that folder. I've been running Sci2 from my downloads folder for about 10 years now, and it's worked perfectly fine. So feel free to put it there. You could also put it in your My Documents, anywhere where you can actually put a file and actually run stuff and save files. Put it there, run it from there, and you're fine. You're good to go. So just some fair warning about the actual Science of Science tool itself. So having said all of that, we're going to now go ahead and jump into the live demo. So with this live demo, we're going to start, again, we're going to pick up with where we left off in the last session with that set of records from the Fogarty International Center that were published from 2011 to 2015. That set of 920 records that Candace went over how to actually do all of the parsing, all of the data cleaning, so on and so forth. So you'll notice here I've got my .isi file. That was the original file that I created, that I downloaded directly from the Web of Science database. I've got my parsed version of that file that I parsed with the Sci2 and saved as a CSV. And I have the cleaned version of that CSV file that I'm actually going to be working with in the actual live demos. So what I'll do is I'll actually demo how to work with either a .isi file directly from Web of Science or from the cleaned version of the file, which is the way that we recommend working. So to do that, I'm going to go ahead and launch the Science of Science tool. Whenever I launch it, I get this warning message about a security warning. The publisher of the application could not be verified. It's fine. Don't worry about it. Just click run. Again, we've been using this for almost 10 years now. We've never had any problems, so just click run. You're fine. So once it actually launches, this is what the Science of Science tool actually looks like. So let me give you a quick overview and sort of introduction to what you're actually looking at here. You'll notice in the majority of the display is taken up by these 4 boxes. The first box here is called the console. This is going to be the place where Sci2 keeps track of all of the actions that you actually take in the Science of Science tool. It's also going to print out a number of warning messages, error messages, any other things that you tell it to print out. So if you try and tell it to calculate a network statistic, the statistic is going to show up here in the console. So this is going to be where most of the interaction with the tool is going to take place. Below that is the scheduler. This basically just keeps track of the algorithms that are actually running in the tool. It keeps track of the date and times they were run, and if they're still running, there will be a scroll or a progress bar that sort of appears down here in the %complete that lets you know it's running. This is the only thing, or the only indication that you will get from the Science of Science tool that it is actually working on something. So it is important to keep track of that, look down at that, and make sure that it's actually done before trying to do something else. Because this is the only time you're going to get any sort of indication at all that it's actually running something. So fair warning. In the upper right is a box called the Data Manager. So this is going to keep track of all of the files that you create in the Science of Science tool, and also any additional modified versions of that file, of those files, are also going to appear in the Data Manager. What is nice about the Science of Science tool is that it never actually touches your original data set, and it never actually creates, and it never actually modifies any specific file that's actually loaded in the data set. So anytime you do anything to a data set or create a network, it actually creates a new file that appears here in this Data Manager. So if you create a network, it's going to appear in the Data Manager. Then if you do something to the network, it's going to actually create a new version of the network with those changes applied. So what that means is you can basically get back to any previous version of a file that you created in the Science of Science tool simply by clicking on the relevant file that appears here in the Data Manager. So there is no undo buttons per se in the Web of Science, in the Science of Science tool, but you can use the Data Manager as an undo feature in order to go back and make changes or undo things that you've done previously. So you can sort of use the Data Manager as your undo feature. The final box down here in the bottom right is the Workflow Manager. So what's nice about this is, again, it not only keeps track of the individual steps that you take, but you can also save a workflow and rerun it later on. So if you establish a series of steps that you're going to run on future data sets, or if you want to save it for reproducibility standpoint, you can use this as a tracking feature to save a particular series of steps in the order that you took them in order to come back to them later. So again, from a reproducibility standpoint, this is great for sort of keeping track of all of that. This is actually separate from that log file that I mentioned earlier. So if, again, you need to go back and sort of redo steps or look at steps that you took, you know, maybe 6 months to a year after you did something and you need to go back to something, you can, again, you've got those files in 2 separate places that you can go back and refer to. All of the actual algorithms and actual steps and workflows that you're going to be working on in the Science of Science tool are in these menus at the top. So all of the actual interaction that you're going to have with the Science of Science tool happens with these menus. Fair warning now, there are a lot of submenus and there are a lot of pop-up boxes associated with working with the Science of Science tool. Most of the time you go to a menu, you go to a submenu, sometimes you go to a sub-submenu, you select a thing, then you get a pop-up box to modify that thing, and then it actually runs. So there's a lot of these kind of menus and submenu operations that happens. They try and organize the algorithms into phases of the analysis. So generally, very, very generally speaking, you work from left to right through these menus. There are a lot of exceptions to that general left to right workflow, but that's the general idea. So you would start over here in File to actually get some data in. You would do any data preparation steps. You would then do pre-processing, creating the networks, modifying it, and then moving through to the visualization steps. Again, there are a lot of exceptions to that, but that's the idea. The other thing to point out about these menus is that it's important to check that you actually have all of these menus in your installation of Sci2. In some cases, if it's unzipped to a folder and it's not working properly, you might not have some of these file menus. So you may open the tool and you may not, in fact, even have a file menu. Sometimes you might not have a data preparation menu, other things like that. If you don't see some of these menus, it's an indication that the location probably isn't correct, or for some reason the installation didn't work. And so you'll need to troubleshoot to actually get the thing to work. So fair warning, that's one of the only indications that you'll have that the installation didn't actually work. So make sure that you actually see all of these menus and you actually can get to all of them. The other fair warning that I will give you is that the science tool, or Sci2, is extremely picky. It is very, very temperamental, and it seems, when you're first learning it, it seems like it breaks very, very, very often. That's totally okay. When I was first learning how to use this tool, it felt like I got error messages or random things going wrong at completely random times. It felt like it broke just because it felt like breaking. So fair warning, there are going to be a lot of things that go wrong when you're doing these things. It is very particular about what you do and the order in which you do them. So if you don't do something in the proper order, it's going to break. If you forget a step, it's going to break. So again, just fair warning, it is very, very picky, and error messages are very common, so don't feel bad if you get lots of them. We all get lots of error messages and lots of things that don't work properly, so just get used to it. It'll be fine. Okay, with all of that said, let's actually go in and start working with this thing. So the first thing that we're going to do is actually load some data that we got from a database. So that's going to happen here in the File menu. This is, in fact, the only thing I ever use the File menu for. There are other things in here, but I have never actually used any of them. So what we're going to do is we're going to do File, and we're going to then click on Load. So that brings up a dialog box where we can navigate to the particular file that we actually want to load. So let me get to that Fogarty International Center set of records. So again, I'm going to start by working with just that raw ISI file, just to give you a sense for what this looks like for going directly from an ISI file. Then I'll talk about how to work with the actual cleaned version of that ISI file that we did in OpenRefine. So again, our recommendation is to work with the cleaned version, but I'm going to show you what it looks like if you want to just work with the ISI file so you know what it looks like. So I'm going to select the file, and then I'm going to click Open. So as it's loading, you'll notice that there's a scrolling menu bar here under the %complete. And then when it's finally done, it will stop scrolling, and you'll see that there are 2 new files that appear up here in the upper right in the data manager. You'll also notice that you've got a whole lot of text up here in the console. Most of the time, that's completely normal. It usually doesn't lead to any errors, but in some cases it might. In some cases, you might actually need to look through some of these error messages or warning messages just to see, because sometimes they can contain clues for what might have gone wrong during that parsing process. But most of the time, you should be good to go. You should be able to just safely ignore this and, again, work with the files that appear here in the data manager. So what's nice about this import process is the Science of Science tool does a lot of things in the background to help you out downstream. So what it does is it parses that original text file into a CSV that we worked with in OpenRefine. It also renames all of the columns. So if you look at the original data set, the columns or the headers are called like AB, TY, PY, other things like that. The Science of Science tool goes through and renames all of those things to much more user-friendly names. So it's a lot easier to see what these columns actually are. It also goes through and creates a pipe delimiter between the individual records, or the multi-valued cells. So that pipe delimiter that we were able to use in OpenRefine was actually created by the Science of Science tool in this import process. So it's going through and looking at saying, all right, it sees 15 authors on a particular record. It just goes ahead and merges all of those, puts that pipe separator between them, and puts them into a single cell that we can work with. So again, it's mostly the Science of Science tool that's sort of doing a lot of that cleanup and pre-processing steps that we were relying on in the OpenRefine session. What you can also do, the final thing that it does is it also goes through and looks for duplicate records. And will automatically go through and remove any duplicates that it finds. So it does that using that unique identification number, or the Web of Science accession number. So it looks for any duplicates in those Web of Science accession numbers, and it will automatically create a new version with those duplicates removed. So if you've got any duplicates in your original data set, don't worry about it, Sci2 will remove them. But interestingly, it does give you the original file as well as the without duplicates file. So again, you have the original file and then a new version of the file with just the unique set of records. And you can go from one to the other simply by clicking on one or the other versions of the file up here in the upper right in the data manager. You can also look at the actual data that's up here. So if you right-click on any file in the data manager, you can either view it or you can save it directly. So you could actually save something, again, as we did in the OpenRefine session. You can save things directly from the data manager to a file. This will actually also work with any networks that you create in the Science of Science tool. So if you've got a very large network that you spent, I don't know, 10, 15 minutes creating in the Science of Science tool, and you want to save that network, you can again right-click on the network, write it out as an XML file, and then that way you can come back to it. You can reload it into the Science of Science tool, or you can load it directly into Gephi. So it's very flexible in that regard. So I wanted to, again, point out some of those things that it does and some of the functionality that's available to you within this data manager. So the first thing, now that we've got all of the records loaded and in memory in the Science of Science tool, we're again just going to click on the set of unique records, and then we're going to actually create a co-authorship network from this .isi file. So to do that, we'll go up to Data Preparation, and we'll select Extract Co-authorship Network. So what's nice about, again, the Science of Science tool is a lot of the networks that you're going to be creating have default values built into the Science of Science tool. So it's designed for actually working with a number of these kinds of bibliometric networks. So it also has functions for creating word co-occurrence networks, creating paper citation networks, author paper networks, all kinds of other kinds of networks that we might be working with in doing bibliometrics. So again, not only is it designed to read Web of Science and bibliometric data files, it is also designed to create networks from those files in very common formats. It's also worth pointing out here that there are generic networks as well. So not only can you create co-authorship networks and bibliometric networks, you can also use the Science of Science tool for any other type of data to actually create networks from any other kind of data you happen to be working with. So you can create these either generic co-authorship networks or generic co-occurrence networks where you're creating undirected networks, or you could also create directed networks from two columns within any data set you happen to be working with. It requires some customization to do these kinds of steps, but I wanted to point out that it is available. So if you're working with other kinds of data and you want to create networks from it, you can actually use the Science of Science tool to create some of these other kinds of networks. But again, in this case, we're going to go ahead and create the co-authorship network. So we're going to select Extract Co-author Network. So what we get is then a pop-up box. So this is the way that Sci2 works. You navigate through the menus, you select an algorithm in the menus, and then you've got a pop-up box to modify the behavior of that particular algorithm. Most of the time, this is kind of the workflow. You select an algorithm, you select the pop-up box, and then it will actually run. So if in some cases you see this kind of scrolling thing and it never seems to end, you may have a pop-up box waiting for you that Sci2 is actually waiting on additional input from you to actually run the thing. So if it hangs for a very long time, go looking for a pop-up box, and that may be what it's looking for. What's somewhat unhelpful is the pop-up box does not actually show up in your taskbar at the bottom, so you kind of have to go looking for it. It's not actually going to create a new window, so you kind of have to go looking for these pop-up boxes to make sure that you actually see them. So again, fair warning, most of the time, go looking for these pop-up boxes. So in this case, we've got a pop-up box that actually asks us what file format we're actually working with here. I really don't understand why we have to do this, because that .isi extension was itself a signal to the science tool that we're working with ISI data, but we have to tell it again that we're actually working with ISI data. So in the dropdown, you actually have to select ISI. Note, however, that EndNote is an option here. So we can, in fact, load data from an EndNote library and then create a co-authorship network from that EndNote library. It requires a sort of special output format from EndNote, but it will work. So if you want to curate a list of publications in EndNote and then move them directly into the science tool from EndNote, that is a possibility. So again, something worth pointing out. But in this case, we're going to select the ISI file format, and then we're going to click OK. It's again going to take a little minute, and then once it's done, you'll see that now we've got this extracted co-authorship network here in the data manager. Again, if we wanted to go ahead and save this, we could just right-click on it and then save it. But in this case, we want to do a couple of additional things and additional steps before we actually get there. So now that we've got the actual network in memory and working with it, we can do some things to see, to get a little bit of information about this network that we just created. So to do that, we're actually going to skip over a couple of menus, and we're going to go to Analysis, we're going to go to Networks, and then we're going to do Network Analysis Toolkit. So within each of these menus and submenus, it tries to organize the algorithms around a particular kind of analysis that we're working with. So it has a lot of things and a lot of algorithms for other kinds of analysis beyond just network analysis. So it's got some geographical analysis options, it's got topical analysis options, it's got a lot of those other things also built into the tool. So it sort of tries to organize what the algorithms are around the kind of analysis you're trying to run. So in this case, we're going to stick with the network set of workflows, and we're going to run this network analysis toolkit algorithm. So what this will do is it will give us a series of network statistics about this network we just created. So it tells us the number of nodes, it tells us the number of edges, it tells us some of the node attributes that it actually has available within this network, some node edge attributes, and some other sort of more network inclined attributes associated with this particular network. This can be pretty handy when we're actually looking at whether or not we need to actually prune this in any way to make a more visually understandable visualization. So in this case, from this set of 920 ISI records, we have 3,600 authors and a little over 60,000 edges. So this is a very dense network. There's a lot of information contained in this original network. In some cases, we're going to actually need to remove some of this information to make it a more visually understandable visualization to actually go to the visualization steps. But again, you can sort of get a sense for whether or not you need to do this from this network analysis algorithm or this network analysis toolkit. It also tells us that we actually have 2 isolated nodes in here, and then we also have 15 weakly connected components. So what does that mean? The 2 isolates are single authors who are not connected to any other author in this actual network. So there are the two isolated authors that are just kind of hanging out. They're just islands. These 15 weakly connected components are groups of authors that are connected to each other within the component, but not connected to any of the other components. So again, if we were just to go to visualize this as is, we would have 15 islands of authors that are not actually connected to each other. So in a visualization, sometimes that's interesting to know, sometimes that's not interesting to know. It depends on the goals of your analysis. So in some cases, you want to visualize all 15 of these islands because you want to make the case that these things aren't actually connected to one another. In other kinds of analyses, we're more interested in just the largest connected component or the largest group of authors that are all connected to each other. So in that case, it actually also tells us that the largest connected component is 3,559 nodes. So 3,500 of our 3,600 nodes are all connected to one another, and then these other 15 groups are maybe 1 or 2 or maybe 4 or 5 authors who are not actually connected to this largest component. This is very, very common in co-authorship networks, where you've got one component that's got about 95 to 98% of the nodes in your network, and then you've got a whole bunch of these other islands of maybe 2 or 3 authors who are not actually connected. So for most of purposes, we just go ahead and remove all of those other connected components and just focus on the set of authors who are all connected to one another. In some cases, we can do this all in one fell swoop. In some cases, we can do this in stages. So one of the things that we can do is if we're just interested in that largest connected component, we can actually remove both the isolates and all of the islands all at once with a single step. So we can actually skip over here to analysis, and this is where it's going to get a little bit crazy in the submenus. So we go analysis, networks, unweighted and undirected, and then we would select weak component clustering. So that would be the way that we would go about doing this. Notice, however, that all of these things are grayed out. And so if I try and click on anything, nothing actually happens. This happens to me all the time and will happen to you all the time. It's a very, very easy fix. What has happened is because we ran that network analysis toolkit, it created a new file in our data manager, and it created this new text file with all of the results of that algorithm. So all of the things that are printed out of the console are also contained here in the data manager. The problem is when you run that, that text file then becomes active. It becomes selected. It becomes the thing that Sci2 is going to try and work on later on. So in this case, because it's a text file, it can't run a network algorithm on a text file. So that's why those things are grayed out. Again, this happens to me all the time. It will probably happen to me again when I try and do things later on in this session. So don't worry about it. It happens. Again, just go back up here to the data manager, select the actual co-authorship network, and try again. So in this case, again, if we go to analysis, networks, unweighted and undirected, we now can actually click on things. So now we'll select that weak component clustering. And again, we'll get a pop-up box telling asking us how many connected components we actually want to create. If we leave this at 0, it will create a new network for all 15 of these components. It's not really what you want to do here, so change this to just the top 1. If we're interested in more, we might say 2 or 3 if we want to, but most of the time we'll just extract this on the top 1 cluster and then select OK. So again, it will create this new network for us with just those 3,500 nodes that were in that largest connected component. So again, we didn't lose that original co-authorship network, and if we wanted to go back to it, we could always go back up to the data manager, select that previous version of the file, and move on from there. So again, every step you take along this process, it doesn't actually lose any information. It just creates a new version of the network that you can work with. So again, if you want to undo anything, just select a previous version of the file, and you're good to go. So with this, again, we could run that network analysis toolkit again just to get a bit more information about this one connected component. And now, so we've got those 3,500 nodes, and now we've got 59,878 edges. So we've got a few fewer edges, but not a whole lot. So again, we just got rid of a lot of those edges that didn't have or a lot of those islands that didn't have a whole lot of authors. In some cases, we might want to go ahead and visualize this network directly, or in some other cases, we might want to prune this even more because, again, 59,000 edges is a lot of edges to try and visualize. So maybe we were more interested in looking at the strongest connections between authors, or maybe we're only interested in authors who have written some number of papers. So there's a couple of different ways that we can go about pruning this network to make it a bit more visually understandable. And in some cases, we want to try multiple approaches. This is where we want to start kind of playing around with some of the things that we get. We might want to try doing both of those things, moving over into the visualization, seeing what we get, maybe trying something else, moving into that visualization, seeing what we get from that, and then making a decision about how we actually want to proceed. Again, Site2 makes this very easy, where we can run one and then just go right back to a previous version of the file, try another one, other things like that. So to show you just a little bit about what this is going to look like, again, we've got a couple options for doing this network pruning up in the pre-processing menu. So again, make sure we've got the network selected, and then we'll go up to pre-processing, and again, go to networks. And then we've got a number of options actually for deleting nodes, deleting isolate nodes, extracting nodes, or extracting edges. So we've got a lot of those options available. The thing that I usually start with is this algorithm called dichotomize, which is going to basically remove edges below a certain threshold. I have no idea, by the way, why it's called dichotomize. It just is. I just run with it. I don't know why. It just is. So I just go ahead and select dichotomize. And so in this case, we're going to get another pop-up box to remove edges below a certain comparison. So in this case, I want to do weight greater than 1. You might want to set this threshold higher or lower depending on how many publications you have, how long the time period is, other factors like that. Typically, I'll start with 1, see what it looks like, and if it's still too big, I might bump that threshold up a little bit higher. It just kind of depends on the network. So I'll start with weight greater than 1, and then select OK. And again, now it gives me this new version of the network where I've only got edges with a weight greater than 1. What I have not done, though, is I've not created, is by removing, or what I've done here is I've just removed edges now. So when I remove all of those edges, I've probably created a whole lot of additional isolate nodes because a lot of the authors in the network are only connected by one co-authored paper. I probably also created a number of islands, a number of other things like that, where I'm sort of, it's a much more scattered network. So after I remove these edges, I want to rerun that weak component clustering to, again, focus on just that largest connected component. So again, we'll go back up to analysis, networks, unweighted and undirected, and then weak component clustering. You'll notice I run this a lot. This is just one of those workflows that just happens a lot. So, you know, it's just one of those things that I get used to. So I'll select weak component clustering, and I'll select just the top cluster, click OK, and now I've got a cluster of 878 nodes. So just by removing that edges of weight less than 1, I've gone from 3,500 authors all the way down to 800 authors. So I've pruned this pretty considerably already. So in some cases, again, that's what you want. In some cases, that's not what you want. It just depends on the goals of your analysis. Another way that we could do this is, again, we could go back to that list of 3,500, and we could just remove authors with a certain number of publications. So maybe we're only interested in people who have written 2 or more publications in the network, but we want to keep all of the collaboration edges there. The way to do that is we would go up to pre-processing, networks, and then, in this case, extract nodes above or below value. So we'll do that. And again, in a minute, we'll get a pop-up box. To select the threshold and then the numeric attribute that we want to actually extract from. So in this case, we want to extract from just 1. In some cases, we might want to bump it up to maybe 4, so we only keep people with 5 or more papers, you know, whatever the goals are, whatever the case may be. But in this case, I'll just leave it at 1. And then for the numeric attribute, we want to change it from times cited to number of authored works, because again, we want to number the number of papers, and then click on OK. So now we've got this nodes above 1. Again, we want to run that weak component clustering just to remove any of the additional isolates. So we'll go up to Analysis, Networks, Unweighted and Undirected, weak component clustering. Select the top 1 cluster. And now we've got a cluster of 990 nodes. So it's similar to the previous one, but it's got a few more authors in it because we're keeping all of those co-authorship networks intact. Or we're keeping all of those links intact. We're just extracting from the nodes the number of works. So different ways of sort of going about this for different purposes. So that's how this would work, again, with an ISI file. I'm very quickly going to just run through what this would look like on a cleaned version of that ISI file, because again, we want to work with the cleaned version rather than the original version, because it does in fact make a difference to our networks and our visualizations. So to load that cleaned version of the ISI file, we're going to again go back to File and then Load. And so in this case, rather than selecting that allrex.isi file, we're actually going to create, we're going to load that allrex parsed cleaned.csv file that we exported from OpenRefine. So once we've got all of that cleaned up, we create that new version of the file, and then we load it in as a regular CSV file rather than an ISI file. So we'll open that. And in this case, we'll need to specify what kind of CSV file we're actually loading. So CSV, the Site2 software is designed for working with data from a number of different sources. So it has a couple of options for the NSF CSV data or the Scopus CSV data. So they're in there. They are options for you to work with, but most of the time we'll just select the standard CSV format and we'll click on Select. So when it's done loading, we'll again see a new CSV file located up here in our data manager. So this is now something that we can actually work with in the Science of Science tool. One note of caution about this is that if you renamed any of the columns from the original data file, you're going to need to follow a slightly different approach. So in most cases, it's a good idea to not rename any of the columns. So if you're working with ISI data, load it into OpenRefine, work with it in OpenRefine, but don't rename anything. Just do all of the processing in the columns with the original column names. If you change the column names, some of the downstream algorithms may not work properly, so you'll need to be aware of that. So just don't touch those original column names, just leave them as is. So if we do have those column names correct, we can actually follow the exact same workflow that we did from the ISI file. So we can go up to data preparation and then extract co-author network. Again, we'll get a pop-up box to select what kind of file format we're working with, in this case, ISI. And then it will extract this co-authorship network. What I'm going to do now is I'm actually going to go ahead and run the network analysis toolkit just to give you a sense of the difference between the uncleaned version and the cleaned version. So with this cleaned version, I now have 3,400 authors instead of 3,600 authors. And then edges, I've got 58 or almost 59,000 instead of 60,000 edges. So we've reduced a number of those edges and the number of nodes by a relatively healthy margin. So we've done a lot, we've reduced a lot of those sort of co-authorship structures and also the number of nodes by some degree that we can actually then work with in the actual visualization. It's also much more accurate. So it does, in fact, make a difference. It also makes a difference when we actually start trying to prune this network. So if we go back in to do some of that data pruning, we can come up to pre-processing, networks, and then dichotomize. Again, we'll do weight greater than 1. And then run analysis networks unweighted and undirected. We component clustering, grab just the top one cluster. And now we've got a cluster of 915 nodes as opposed to the weak component cluster of 878 nodes that we got from the uncleaned version. So in this case, we actually have more authors retained in that in that largest connected component because we did a lot of this mean cleaning we associated with for different variants, and so the connections between authors are somewhat stronger, so we actually see more authors in this pruned version of the network than we did from the uncleaned network. So it does make a difference. So this is, again, why we recommend that we want to go ahead and do this mean cleaning before we actually go off and do additional analyses and visualizations. If we want to go ahead and save this particular version of the network, let me just show you what that looks like, because you can't actually save work directly in the Science of Science tool. You have to actually save individual files that you create individually. So again, you can save this workflow if you wanted to, but most of the time if you're saving files, you actually have to do that through the data manager. So to save this particular version of the network, we right-click and then click on Save. So this will bring up sort of a pop-up box that asks you what file format you want to save this network into. There are a couple of other file formats. NWB is a file format that's proprietary to Sci2. It's nice if you're only working with Sci2, but nothing else can read that file format, so it's relatively of limited value otherwise. There are a couple of file formats for a tool called PIAC, which is a computer science network analysis tool, very, very common in computer science. So if you're interested in working with it in PIAC, you can do that. Most of the time, however, I leave this alone and save it as this GraphML file. So essentially what this is an XML file that describes the nodes and the network and the edges in this particular network in a very generic file format. So virtually any other network visualization software, network analysis package can read GraphML files. So virtually anything else can work with this. So it's much more flexible in terms of moving things to different tools. So I always keep it as GraphML, and then we can go ahead and select, and then we can select the file format, change the file name, the file location, other things like that, and actually save the file directly as this .graphml file. So that would be the way that you would actually want to save things is, and particularly saving networks, would be to save them as this GraphML file format. So the other thing that we can work from here is we can actually go directly to Gephi from the Science of Science tool. So once we have this co-authorship network created, we're happy with the way that it looks, we can actually create a bridge directly into Gephi from this visualization menu. So as long as we've got this particular network highlighted, it will launch that directly in Gephi by going to Visualization, Networks, and then Gephi. So again, once you select that, it will load this, it will open Gephi automatically and load that particular network file directly into Gephi. So it's a very seamless process. This is also why we tend to use Gephi over a number of the other visualization software tools, simply because it's built into the Science of Science tool. There are some other plugins for other visualization software. If you're more comfortable with using Cytoscape, for instance, there is a plugin for that, but most of the time we'll just use Gephi and then go from there. So That's kind of the generic workflow that we'll typically use for individual level author networks. Again, we can work either from the ISI file or the CSV files that we happen to have. Another thing that we can do is we can start creating some more customized versions of networks. So in this case, we can also work with that cleaned CSV file to extract a co-occurrence network on the research addresses or the organization names that we did the name cleaning on in OpenRefine. So if we're going to do that, that requires a little bit of a customization. So in order to have all of the attributes present that we had in the co-authorship network, we have to do a little bit of a detour. We also have to do some of that name cleaning from the original Web of Science file because those files had those author names and all of that additional address information in the individual author records. So if you remember back to the OpenRefine session, when we were looking at the research addresses as they came out, we had all of those authors in brackets. We also had like the street addresses, we had the countries, we had the cities, we had all of that information. And with all of that information, we can't really extract a network based on that because it doesn't necessarily work within the tool. We actually have to use the cleaned version of the ISI file in order to actually create this. The other thing that we need to do is, again, in order to have the kinds of attributes associated with this particular network, we need to use what's called an aggregate function file to customize the way that Gephi actually works with things and how the Science of Science tool actually creates networks. So let me explain what those things are and give you a little bit of a sense of what I'm talking about. So aggregate function files are external text files that you can write to actually add additional attributes to a generic network. So if you're going to create a network from your own data set or create a network that Sci2 isn't expecting you to work with, you need to tell it what sorts of attributes you need to calculate in order to actually have those attributes present in your final network. So remember, in the network, we had the number of co-authored works available, we had the number of authored works available, we had the time cited counts. We had all of those attributes present in the co-authorship network that we could use to filter later on downstream. If we just try and create a generic network from a regular just CSV file, we won't have any of those attributes in the final network. So we need to tell Sci2 what attributes we want to calculate and how to actually calculate them. So that's what these aggregate function files are. So again, they are these text files that sort of live outside of the Science of Science tool that you load in when you're creating networks to tell it what to do. In this case, I actually have a folder full of them because I customize the Science of Science tool all the time. So I'm going to scroll down to the particular one that we're going to be working with, which is this research_address.txt. So let me just open this up and give you a sense for what this file looks like. Again, it's just a flat text file that's hanging out on my computer. And within that text file, I've got these 3 lines. And so these are sort of extensions of a Python-like code that tells Sci2 what attributes I want and how I want it to actually calculate those attributes. So in this case, we need to specify first what kind of attribute we're dealing with. So in this case, we're doing a node attribute, so we do node dot whatever the name of the attribute is going to be in the extracted network. So I've got node dot number of works. So create a node attribute called number of works, create a node attribute called citation count, and create an edge attribute called co-authorships. So those are the 3 things that I'm telling Sci2 to do. Then in the other side of the equal sign is how I wanted to actually do that. So in this case, I want to take values from the research addresses column in the data set and then I want to tell it to count what it finds in that column. So in this case, we're going to count the appearances of each node in the research addresses column, and since the rows indicate the number of authored works, we're going to assign that just the number of works. So we're going to count up the number of times an organization appears in the data set, and we're going to call that the number of authored works. So that's essentially what we're doing. So we're going to create a node attribute, call the thing, we're going to get it from a column, and we're going to do a thing to get it. That's essentially how these files work. So we're going to do that. We're also going to do node.citation count. We're going to create this citation count attribute. We're going to get it from this time cited column. And in this case, instead of counting it, we're going to sum. We're going to add up the numbers that you find in the time cited count. And so that'll give us a total number of citations for each one of the research addresses in our data set, in this particular data set or publication set. The last thing we're going to do is we're going to create an edge attribute, which is going to be the co-authorships. So it's going to be the number of co-authored works. So we're going to get that again from the research addresses, and we're going to count the number of times. So essentially what we're going to do is we're going to again count the number of appearances of each research address, but we're also going to count the number of times each research address co-occurs with every other research address in the data set. And so the result of that is going to be the number of publications where those two research addresses have co-authored works. That's essentially the idea behind these. You could again create all kinds of additional attributes using this general format, or you could simply just reuse what we're seeing here and just go with it. The thing about these aggregate function files is they are extremely picky about the column names and the format of the column names. If you don't capitalize both research and address, it will fail. If you capitalize one of them but not the other, it will fail. If you say just research address, not research addresses, it will fail. If you have an underscore instead of a space, it will fail. On and on and on again. So this is again why I said if you're creating co-authorship networks and you want to use those cleaned versions, don't touch the column names. Leave them exactly as it is in order for this aggregate function file to actually work properly. Because again, if it doesn't have that exact column name, it's not actually going to work properly. So be very, very careful about that. So that's a bit about aggregate function files. So let's actually see what this looks like. So what we're going to do is we're going to go back to Site2, we're going to go back to the data manager, and we're going to go back to that clean version of the CSV file that we loaded in a little bit earlier. And then we're going to again go up to data preparation to create the actual network. But in this case, rather than creating the extract co-author network, we're actually going to extract a co-occurrence network. So we're going to use one of these generic functions that's built into the tool using that aggregate function file that we use to customize the actual way that it works. So we'll do extract co-occurrence network. And so this time the pop-up box looks a little different. We actually need to tell it what column we want to actually extract the network on. So from the drop-down, we need to scroll down to research addresses, which is the file, which is the column name that we want to actually extract it on. The text delimiter, we need to specify whether it's a pipe delimiter, semicolon delimiter, or other things. Again, because most of the time we're working with this workflow from Site2, that pipe delimiter is what we used. If you rejoined the cells using a different delimiter, you need to put that delimiter in here or else it won't work. So again, it's best practice just to stay with that pipe delimiter because that's what Site2 is expecting to see. And so the final thing that we have is this option for that aggregate function file. So this is where we actually browse in and grab that file to tell it how to run. So you can click on Browse and then navigate to that file, that research addresses.txt file. So in this case, I'm going to go back to my folder full of aggregate function files. I'm going to grab the research address.txt file and then click on Open. And so that way it will then run all of the commands that it found in that particular file, and then click on OK. So now it has this extracted network on column research addresses. So again, we can do analysis network and network analysis toolkit to get a little bit of information about this network we just created. So in this case, we've got about 1,000 nodes, we've got 15,000 edges, and then we've got some of these attributes associated with them. So again, we've got with the node attributes, we've got the label, we've got the number of works, and then we've got that citation count. Again, that came from that aggregate function file that we ran. So from this point, we can then go ahead and do some of the similar kinds of preprocessing steps that we did in the previous version. So we could, again, want to clean this up a little bit, remove some of these edges to focus on just the largest number of co-authored works between institutions. So we can, again, go back to extracted network. We can do, again, just like we did with the co-authorship network, we can run all of the same things on this new network. So we can do preprocessing, networks, dichotomize. We can do weight greater than some threshold. In this case, I'm actually going to bump this all the way up to 9. So I want to keep co-authorships with 10 or more papers between organizations, because the organization collaborations tend to be much higher weights than the individual level authorships are. Again, we're looking at all authors from these institutions that are collaborating with any other author from another institution, so the number of co-authored works is generally a lot higher with this level. So I'm going to set that threshold a lot higher this time. I'll say weight greater than 9. And then again, analysis, networks, unweighted and undirected. We component clustering just to pull the top cluster of nodes. And so now we've got a cluster of 142 nodes. So this is probably what I would go ahead and work with in the actual visualization settings. I might play around with setting this threshold a little bit lower just to see how many nodes I get, how many edges there are, just to sort of play around with that. I might even go ahead and visualize something, see what it looks like, and if it's a giant hairball, then I might come back, set the threshold a little bit higher, and then visualize that higher threshold. So again, there's some playing around involved with sort of where you set these thresholds based on the data set, the analysis that you're working on, and the goals that you have. So you might kind of go back and forth, play around a little bit, see what works, and then kind of go with what makes the most sense. So again, that's we can then go ahead and go up to visualization, networks, and then Gephi to actually go into the actual visualization steps. So that's all that I wanted to cover for this particular course. Again, I wanted to just kind of give you an introduction to network science, a little bit about the workflows that we can work with within Sci2 to create networks, to prune them in various different ways, create custom networks, and so on and so forth. We will be returning to the aggregate function files in the topic analysis course because we use those extensively in our workflows around topic analysis. But then in the meantime, in our next course, we're going to pick up with these co-authorship networks and talk about visualizing them in Gephi.